So with this, I have the pleasure to invite the last speaker of this session to the stage, which is Leif Sander from the University Hospital Charité in Berlin. He studied medicine and is a medical professor. After studying in Hanover and doing his PhD there, he traveled the world a bit, was in Aachen, in New York, and then finally ended up in Berlin. He is a professor for infectology and the director of the medical clinic with a focus on infectology and pneumology. I should know better. We will stay in the topic of COVID-19, and Leif will talk about myeloid cell responses in severe COVID-19 patients and the implications for other lung diseases. So welcome here, and the stage is yours.
Well, thank you very much for the kind invitation to this conference. I have to warn you that I do not know very much about drug delivery, and I'm not primarily going to be talking about drug delivery, but I'm going to be talking about the cell types in the lung that are mediating damage in severe COVID and what we think their role might be also in other lung diseases, and maybe you have ideas on how to target them. So as a respiratory disease physician and an infectious disease physician, when first cases of this unknown severe pneumonia in Wuhan, China appeared in the news, we as a big university center, we decided that we needed to set up a study, and it became very obvious that probably this new virus was going to spread across the globe, because as you can see here from the very early days, the dynamic was very different than, for example, the SARS outbreak in the early 2000s. And what we decided to do as a medical center, so we're supposedly the largest university medical center in Europe, and we do serve a very big population in Berlin. We decided to set up a phenotyping platform in which we would enroll all the COVID patients admitted to Charité, be it on the floor, on the ICUs, and to perform a very detailed and granular, both clinical as well as experimental and exploratory phenotyping on these patients, since this was a new disease that we didn't know at the time. And so we've enrolled something in the neighborhood of 1,300 patients. So we stopped at some point, and this was actually transferred to a national platform, and we wanted to look into risk factors that determine poor outcome or severe disease in COVID, and features of protective immunity, and also develop treatment and prevention strategies. So as clinicians, what we had to learn early on was that this disease sometimes comes in several phases, in a biotriphasic disease. So, and what's typical about COVID and other viral diseases as well, but particularly COVID here, was that at the stage when the patient is infected but not yet symptomatic, you have very steep and very rapid viral replication. And at the time when the first symptoms appear, around that time, that's when you reach peak viral loads, and that's probably why this is so good at spreading, because people are not aware that they're already infectious and they can pass on the virus to other individuals around them. And then within 10 days or so, the symptoms would recede, and we would see seroconversion. So this is something we picked up very early, that by day 10 or so, most patients would have detectable IgM and IgG antibodies in their serum. And it's only in the second or in the third week that the proportion of those patients would deteriorate and show up then on our wards or on our ICUs with respiratory failure and ultimately ARDS, which we just heard about, and critical illness requiring mechanical ventilation or even organ replacement therapy such as ECMO treatment. And we had a lot of those patients on ECMO through the different waves that we experienced in Germany. And these were some of the first images that came, actually were reported from Asia, but these are very typical radiographic images from patients. You see these diffuse infiltrates in the early stages, these with ground glass opacity, so indicating a disrupted alveolar barrier and maybe early infiltration of immune cells. But then later on we can see more and more consolidating lesions and this full-blown forms of ARDS that we can see here. So unfortunately, on our ICUs, about 50% of patients, we had really very sick patients on our ICUs that were on ECMO, and we had a mortality rate of about 50%. This is not only a very heavy burden on the personnel on these wards, but also it opened up an opportunity for us in an autopsy study to basically look into the lungs of what was going on or what was going wrong in these lungs. And what you can see here, this is a control histograph of a normal lung as you know it, this very delicate, fine alveolar architecture. And this is a lung of a patient who died from severe COVID. This was one of the first patients that died on our wards. He came from a skiing vacation and unfortunately didn't survive this. But what you can see here is diffuse alveolar damage, hemorrhage, and massive infiltration of inflammatory cells. And particularly what we noticed very early on is that you have very dense infiltrations of macrophages and other myeloid cells. And to better understand what was going on in the innate immune response, or in the myeloid cell compartment, so in the granulocyte, monocyte space, in patients with different forms of COVID, we set up a dual center study teamed up with colleagues from Bonn and sampled patients longitudinally, so in the early stages, in the later stages of the disease, patients that would not progress to severe COVID and others that experienced severe COVID, and some of which also deceased. And we basically subjected blood samples and peripheral mononuclear cells to multi-omics, single-cell multi-omics techniques that would allow us to get a very highly resolved picture of the myeloid cell response. And I'm going to not show all the results here, but this is just showing you how you can, if you run single-cell RNA sequencing on peripheral blood mononuclear cell, you can really get a very nice resolution of the cellularities in those patients. And if we now stratify the patients or categorize the patients by severity, these are the cells that were basically derived from patients with mild disease and those derived from patients with severe disease. What you can immediately see that there's a population of cells that is present in the patients with severe disease that's absent in patients with mild disease, and that also the distribution of cells in this space here, and this is the monocyte space, is very different. So this is, of course, a way to reduce all the dimensions in the transcriptional signatures that we see, but through the distinct distributions, we can see that the transcriptional patterns are very different. And indeed they are. If we zoom in onto the monocytes, we can see that we've color-coded them here, so we have these purple clusters and these green clusters. What you can see here that in patients that basically have a moderate course of the disease and they recover just fine, they have these green clusters, and the more severely ill patients, they develop these purple clusters, and some of which they're characterized by low expression of MHC molecules. So they're probably not very good at presenting antigen, but they produce a lot of other factors such as alarmins, which are pro-inflammatory, and also this marker CD163, it's a scavenger receptor, which I'm going to come back to. So if we basically, what we found here as one of the first groups, so was basically that dysregulated myeloid cell compartment really is a hallmark of severe COVID, which was confirmed by many other groups as well. And if you look into monocytes, for instance, from patients that make it well through the disease, maybe require a little bit of oxygen, they are appropriately activated. As you would expect, many molecules that they express would be expected in viral infections, versus in patients with really severe or critical disease. Those monocytes are dysfunctional. They don't activate T cells very well. in a normal fashion. And they also have signs of emergency myelopoiesis, so you have these immature forms of neutrophils that you can detect in the blood, but that's a common feature also seen in other forms of sepsis. Now, this was a good lead, but we obviously wanted to better understand what is happening locally, what is happening in the mucosa, in the upper respiratory tract, as well as the lower respiratory tract. And if you remember, at least in Germany, the swabs that we used in the beginning for PCR sampling, they were quite big and they were actually quite nasty. But an advantage of those was that you could actually recover a lot of cells from those swabs. Obviously, they were good because cells and debris that contained viral RNA was basically taken out of the mucosa, but also cells, and we could run those cells when we used some mycolytic agent. We could basically dissociate the cells and also subject them to single-cell RNA sequencing. And what you can see here is that you can really resolve the entire cellularity of the upper respiratory tract mucosa, even detecting some very recently discovered cell types and rare cell types in the mucosa. So this is a very actually nice technique. And without going too much into detail, what we noticed here again in patients with severe COVID, you can basically see that non-resident macrophages that have been recruited to the mucosa, they are strongly activated in patients with critical disease, and this is in the nasal mucosa. And through bioinformatic analysis, you can predict that these cells engage with a lot of cell-cell interactions with all the other resident cells that are non-myeloid cells, also mesenchymal cells and epithelial cells, that they probably orchestrate the mucosal immune response in those patients with severe COVID. One other question that intrigued us, obviously, one thing we were afraid of was what's going to happen to our kids, right? Once this virus comes, it's a new virus. And so we teamed up with our pediatricians at Charité, and we decided, let's compare COVID in kids and adults. And it became apparent quite early on that the kids are actually quite resilient, quite resistant actually to severe. Very rarely do we see severe COVID, and almost never do we see COVID ARDS in those little kids. And so what we did is we performed a study in which we went home to kids that had reported to their pediatrician, and we sampled kids and their parents and compared basically the local immune response in the mucosa of the parents or the grandparents and the kids. And what we found was quite maybe not so surprising, but maybe reassuring, is that kids, the younger the kids are, the more immune cells, certain immune cells, are already present in their mucosa, even at steady state. And if I look at my kids, when they were in kindergarten, they had a runny nose all the time. So I'm not too surprised to see that they actually have a somewhat pre-activated mucosal immune response, these kids. And there are other features about kids' results that we haven't published. They tend to have more certain types of immune cells in the blood as well and produce more interferon, so sort of more ready to go to fight viruses than maybe some of the adults are. And this changes somewhat around puberty. And so when you basically dig in or dive deeper into the data, you can see that even the epithelial cells in your mucosa, they express more molecules. I'm not going to go into details, but these molecules are involved in sensing the virus and making interferon and other factors in response to the virus. And they appear to be upregulated in different epithelial cells here, both in steady state, which you can see here. It's a bit higher expression compared to the adults, but it's particularly upon infection, these cells are really ready to go and express a lot of these markers. So the current hypothesis here is that maybe the kids, they respond very vigorously to the virus. They can get infected just fine, and they also shed the virus and they can infect others, but somehow the virus is not allowed to travel to the deep lung and probably doesn't cause, or very infrequently, very rarely only causes lung damage and respiratory failure in kids. But coming back to our sick adult patients, the ones that were dying on our ICUs, we wanted to better understand what's happening in the lungs. And again, we teamed up with many different teams from our center and other centers across Germany. And what we did was we sampled both bronchoalveolar lavage fluid We again went to our autopsy cohorts, which had been established at different sites in Germany, and looked into the tissue and also extracted the nuclei from the tissue and performed sequencing on those, as well as a lot of clinical characterization and imaging. And again, what we saw was that what I'd shown you before, again, those lungs from those COVID ARDS patients is completely disrupted lung architecture. There's lots of macrophages, but if you look more carefully, all of those macrophages co-stain for this marker CD163, which is a scavenger receptor, which I'd also shown you was expressed on monocytes in the blood. And if you know a little bit about lung macrophages in lung diseases, CD163 positive macrophages are thought to be somewhat alternatively activated macrophages. And they've been shown, for instance, in pulmonary fibrosis and in other diseases to be promoting the disease or associated with poor outcome of the disease. And you can see that they accumulate to a large extent in the lungs of patients with COVID ARDS. Now we can zoom in, again, using single-cell RNA sequencing, we can really resolve the cellularity of those macrophages that we find in the lung, this time in the BAL. And again, we can see that you have monocytes that come in, so this is like a pseudotime gradient. You can model the differentiation of these cells. So you have monocytes that come in from the circulation, they're quite inflammatory, and then very rapidly they differentiate into a macrophage cell type that is somehow arrested in this stage, that expresses CD163 and a bunch of other markers, and you might not be familiar with that, but if you look at those markers, it's really a combination, and I would say this is a cell type that is very good at taking out dead cells, which is good, scavenger receptors. It's also good at interacting with matrix and also stimulating the production of matrix. These are cells that we find in fibrosis. These are also cells that we find in atherosclerosis, and also cells that we find in the vicinity of tumors. So some of these markers have been found in other disease models. Now, as a pulmonologist, not only the macrophages, but also some of the CT scans reminded us of diseases such as idiopathic pulmonary fibrosis, a devastating, incurable disease that affects primarily older males or older people, but there is a higher frequency, a higher incidence in old age and in the male sex. And this is somewhat of an overlap with the risk factors for developing severe COVID as well. Now, and this is just a radiograph, a CT scan of a patient with IPF. Now, if we went to other studies that have been published on patients with IPF, and other groups had run single-cell RNA sequencing on macrophages from patients with IPF, and it was already known that CD163 positive macrophages are correlated with poor outcome in IPF. We can extract those signatures that they found in IPF that are specific to IPF and sort of project them onto our cells that we find in COVID. So this is acute infection and a chronically progressive interstitial lung disease that's not thought to be caused by infection. And what you can see, there's a very high enrichment of those cells in our CD163 population, and you can use other statistical methods to sort of show the same thing. This is to say that these macrophages that we find in the lungs appear to be profibrotic because they share a lot of features of macrophages that we find in IPF. And we can use even more fancy techniques, which I don't understand, but our colleagues at the Helmholtz Center in Munich, they can integrate different data sets from different diseases and then basically integrate them into one data set and then determine the proximity of those cells. And long story short, CD163 positive macrophages map very closely to cells derived from patients with IPF, but not from healthy donors, not from other diseases, while the alveolar macrophages that we find in the late stage, when the patient is recovering and the macrophages are reconstituting their niches, then they again look very much like alveolar macrophages from healthy individuals. If you then look into the tissue, these are unfortunately patients that died from COVID. This is a patient that died from another cause, and what we stained in green here are mesenchymal cells, so fibroblasts and myofibroblasts, and you don't need to be a pathologist to see that you have a massive expansion of these green cells, so it's a lot of proliferation of fibroblasts and interspersed with these red macrophages that are in very close contact. Now, it's been known and been sort of debated for quite a while that the macrophage fibroblast, myofibroblast interaction It's a very dynamic interaction, but that's something that can drive fibrosis and keep fibrosis very active. And fibrosis is one of the diseases that we normally cannot revert. And now when we again run single nuclei sequencing on nuclei that were extracted from those lung tissues, we can again use prediction bioinformatic models to predict that those macrophages indeed communicate a lot because they express a lot of ligand receptor pairs and have other properties that suggest that they interact a lot with fibroblasts and myofibroblasts. So do our patients actually develop fibrosis? And when you talk to intensivists, you will hear that indeed they do. So this is a healthy lung scanned on the CT, and this is sort of the first CT scans that we get once they are intubated in our ICU. You see the typical features of COVID ARDS, but as you progress, you see these consolidations, and then you see these massive fibrotic lesions, though this patient unfortunately didn't survive, but this is very similar to what I showed you on the CT scan from the IPF patients. And again, talking to the pathologists, if you stain tissue for collagen and other, matrix components, the tissue is really full of collagen. And so this is a specific form of ARDS, which is called fibroproliferative ARDS. So this is sort of I think a wound healing response has gone awry, and it's associated. We see it with other infections as well, but not as frequent as with COVID. In influenza, you can see a fraction of patients also developing fibroproliferative ARDS. It's associated with very poor outcome, and you can imagine that we have to ventilate or keep those patients on ECMO for a very, very long time to give them time to recover. And during that time, you can easily catch another superinfection, and that's it. And you can also see ultrastructural changes that are very similar to what we see if we give the pathologist IPF samples. So somehow we see a profibrotic response that appears to be driven by macrophages. And the question was, and I'm just going to cut this short, how is this induced? So for one, if you have massive damage, if you have cell death, it's well known that when macrophages eat up dying cells, they start to produce TGF-beta and other factors that can promote fibrosis. But somehow we performed an in vitro experiment, and you can take monocytes from a healthy individual and incubate them with the virus or with ligands that are meant to mimic viral infection. And you can see that only with the virus we can strongly induce a signature that again makes the cells look like a profibrotic macrophage. So there seems to be, and we can confirm that with other methods, and we can extract different signatures from the literature. So there seems to be something about the virus that induces this pro-fibrotic transcriptional phenotype, which might be a good thing in most of us, but might not be well controlled in patients at risk. And this, by the way, is not induced by influenza virus and other viruses, which we also did. So to sum up this part of the story is basically that upon viral infection, we do get, as we heard, we get infection of the epithelium, we do get disruption of the alveolar barrier, we have leakage of fluid from the vasculature into the alveoli. That's what we see, these ground glass opacities, and we have influx of these pro-inflammatory macrophages, but somehow these seem to be taking up a lot of the virus. We can also see that most of the viral particles after a while reside within the macrophages, and they become pro-fibrotic, and then they start interacting with those myofibroblasts and fibroblasts, and then we get this fibroproliferative response. Now, the final word. So the patients that actually survived severe COVID, that were on ECMO or ICU, they show a remarkable capacity to clear those fibrotic lesions, to also clear those consolidations. And this is obviously something that we never see in patients with chronic, fibrotic diseases of the lung, like IPF. So this is obviously a holy grail. How can you revert that? And how can you reprogram those cells to basically stop producing matrix and maybe start clearing matrix? And this is something that we're doing or have done and sampled longitudinally patients once they come in, and then start sampling in that window to see when does that switch occur and what are the signatures. So we've also initiated a clinical trial trying to inhibit the migration of monocytes into the lung using a chemokine receptor antagonist. And I'm very curious to hear whether anyone has ideas on how to target macrophages in the lung. So I have to thank my group and especially Daniel and Sophia and Anna, who performed all the work together with this is a massive team effort across Germany, and there's lots of funding here from public agencies, and I thank you for your attention.
Thank you very much, Leif, for this very interesting insight into details of COVID-19 infection. Are there any questions from the audience? While you're thinking, maybe I take a question from Slido, and that kind of refers to how to do this holy grail of reversion and getting the fibrosis away. The question here is how to actually turn an adult in a child in terms of their mucosal immune response. So is there any pharmacological or maybe lifestyle-related modification possible?
So there have been ideas around that for quite a while, of sort of administering certain ligands to the mucosa as an inhalation, for instance, in patients that are ventilated on the ICU to prevent the onset of ventilator-associated pneumonia. And these can be TLR ligands, some of which are also, mimic viruses. And they are meant to sort of prime your innate immune system in your mucosa and sort of ready it for any infection that might then come. And what we also know, if you've just had one viral infection, you're less prone to get another one. So there is something as an interferon signature or an antiviral state that you can induce that lasts for a certain time. So there are a number of thoughts around that, but there are really interesting changes in the immune system of kids that really, upon adolescence, that really changes. And I don't, of course, there's got to be some hormones and so on that are involved, but it's really not that clear how this actually occurs, and if we can better understand that, obviously, that would be interesting.
Thank you. There is a question.
This is really fantastic work. You said if anybody could tell you how to target macrophages, that's really, it's always the problem for us drug delivery people, how to avoid the macrophages. If you just put it in a particle it's not immediately dissolved, you can have all your particles in macrophages within a half hour. So I think, but targeting specific populations of macrophages, I don't know about that. All I would find is that all of my particles were in all of the macrophages almost immediately, microscopically.
Yeah, I mean, that's in a healthy lung that is ventilated and it has the resident alveolar macrophages that are very good at clearing particles and anything in a very sort of silent manner without causing damage. Now, I showed you the lungs. There's not much ventilation going on in those most severe patients, and we probably have to have a more systemic way of targeting the macrophages. Again, of course, macrophages are great at taking up particles, and again, these guys take up a lot of the virus, but sort of ways of getting rid of those would be great, and specifically targeting them, of course, as surface receptors. But I doubt that you could give something via inhalation in that late stage. Maybe at an earlier stage that would still be a meaningful approach when the alveoli are not completely flooded yet.
So thank you very much again, and thank you to all the speakers of this session. We now come to an end for today's program, and my co-chair already ran away. I'll follow him.
So today, what have we heard about? Well, we've talked about drug delivery, the past, the present, and the future, and then we had a great session on planetary health, and then Regina, and we've just done the session on lung diseases. So tomorrow, what are you looking forward to?
I guess we're going to have the splendid Pat Burnell session, of course. This is my personal favorite. And there will be a lot more interesting sessions and interesting discussions. But to be honest, for the moment, I'm far more looking forward to something else. And this is the drinks reception. You're welcome.
Have a great day, everybody. See you tomorrow.